Alright, hi everyone, and this is another video. This time we'll be talking about the different ways by which plants are able to fix carbon dioxide. So the title of our module 2.4 would be Alternatives to Photosynthesis Pathways. Now, here's a question. What will happen to plants when it's too hot? And this is very familiar with us. We all know that plants will eventually wilt and will eventually die, so that plants lose water through the stomata, and this is a process called transpiration. And as mentioned a while ago, too much of this, the plants will eventually die. So the question is how to survive. Well, the plants survive by closing their stomata in very hot temperature in order to prevent water loss. But there is an accompanying problem, and the problem would be they won't be able to use carbon dioxide, because since the stomata would be closed, not only will the water vapor be unable to go out of the plant, thereby saving the plant, but the problem is the carbon dioxide can't also enter through the stomata, which thereby causes photosynthesis to eventually stop. So let's look at the overview again. So dehydration is a problem for plants, especially in very high temperature. On hot, dry days, plants essentially close their stomata in order to conserve their water. However, this limits photosynthesis. The closing of stomata reduces access to carbon dioxide, but it causes another problem, and that is the buildup of oxygen. And the buildup of oxygen leads to a process which we call as photorespiration. So that leads us to a question: what is photorespiration? Now, photorespiration happens in high temperature, especially when carbon dioxide is very low. In normal conditions, the rubisco, which is the enzyme, would cause the RUBP to catalyze with carbon dioxide, and this proceeds towards the Calvin cycle, so which we have discussed last time. But the absence or the lessening of carbon dioxide causes rubisco to be attracted more towards the oxygen, so that the oxygen now would be catalyzed by the rubisco to attach to the RUBP, and this is called photorespiration. And photorespiration, as it adds oxygen instead of carbon dioxide in the Calvin cycle, well, not only does it produce a 2-carbon compound, but it also releases carbon dioxide, but without producing ATP or sugar. So this is a problem, because the plant will now be using up in organic molecules that they have already produced, but without producing ATP. So it's a loss for the plants if ever photorespiration would occur. So the question now is, what possible adaptation can solve the problem on photorespiration? So photorespiration, the Calvin cycle, this would be the problem of many plants. So how are they able to adapt to this? So there are three ways by which carbon dioxide can be fixed, and two of those ways would be adaptation for high temperatures, so that the plants will not undergo what we call as photorespiration. So let's look at the three pathways. The first pathway is called the C3 plants, and take note that the C3 will be pertaining to a 3-carbon molecule. So this is the normal Calvin cycle. So the carbon dioxide immediately enters the Calvin cycle and converted to a 3-carbon molecule inside of the Calvin cycle. As you can see here, that the carbon dioxide, as it is catalyzed by rubisco with ribulose 1,5-bisphosphate, it immediately becomes a 6-carbon molecule, then broken down to a 3-carbon molecule. So this is the 3C, the 3-carbon that it pertains to. So this is the normal Calvin cycle that we know of. But of course, the problem is that they are not adapted to high temperature. So the next two pathways will show us two ways by which plants or different groups of plants are able to adapt to high temperature, and that is what we call as, the first one is the C4 plant. So this is the second alternative pathway to carbon fixation. So let's look at how the C4 plants adapted itself to high temperature, and it does this by first fixing the carbon dioxide to a 4 carbon molecule. So it's interesting, compared with the C3, the carbon dioxide is fixed into a 3 carbon molecule, but for the C4 plants, it is fixed into a C4 or to a carbon 4 molecule. And that carbon 4 molecule, as you can see here, the PEP, which is phosphoenol pyruvate, a 3 carbon molecule, will now react by Rubisco to carbon dioxide. So the carbon is added to it, it now becomes a 4 carbon molecule. So this is how the C4 adapted itself to high temperature in order to reduce photorespiration. Since the carbon dioxide is already inside of oxaloacetate, it will simply transform to malate, and then it will donate the carbon dioxide towards the certain cells in order to increase the concentration of carbon dioxide. So that's the most important thing, which is the formation of a 4 carbon molecule. Okay, on the other hand, the third one, CAM plants, their strategy is almost the same. Carbon dioxide is also fixed into a 4 carbon molecule, but the difference is for the C4 plants, it will fix it into a 4 carbon molecule in a mesophyll cell, and then it will release the carbon dioxide in the bundle sheath. But on the other hand, the CAM plants will fix the carbon 4 or fix the carbon dioxide in a carbon 4 or 4 carbon molecule during nighttime, and then it will release it during daytime. So let's look at the carbon 4 first. So you can see here that it's the same process just like in the C4. Phosphoenol pyruvate, which is a 3 carbon, will now react with carbon dioxide, and it will produce a 4 carbon molecule. It's still the same, it is oxaloacetate as well. So that is how the CAM will be doing it. But of course, the difference is the CAM plants fixes the carbon dioxide in the 4 carbon molecule at nighttime, and then releases carbon dioxide at daytime. So for the C4 plants, it's a separation of space, different types of cell. But for the CAM plants, it will be a separation of time at nighttime and also at daytime. So let's just describe these plants. So the C4 plants, again, take note, the most important thing, fixes carbon dioxide into a 4 carbon molecule. There are 2 distinct cells in the C4 plants. We have the bundle sheath and also the mesophyll cell, as you can see over here in the picture. So you will see that the mesophyll cell is over here. This is where the fixation of carbon dioxide into the 4 carbon molecule happens, and then the bundle sheath is where the Calvin cycle will be happening. Okay, now other things we have to take note of, the sugar production happens in 3 steps. So there is a different enzyme called the PEP carboxylase, the phosphoenol pyruvate carboxylase, which is actually has a higher affinity to carbon dioxide, so it is able to absorb the carbon dioxide or catalyze the carbon dioxide more efficiently than Rubisco does. So this 4 carbon molecule will then be exported to the bundle sheath, and then it will be released to the carbon dioxide. Carbon dioxide will be released to the Calvin cycle. So let's go back a little bit to the figure again. So what's very interesting here is that since the mesophyll cell surrounds the bundle sheath, what happens here is that the carbon dioxide will be fixed in each of this mesophyll cell, and then the 4 carbon molecule will then be transported to the bundle sheath over here, thereby releasing carbon dioxide. And what's interesting, since all of the 4 carbon molecule will be going here and releasing the carbon dioxide will be very highly concentrated in this area, which causes increase in concentration. Thereby, oxygen will not be able to really go through it, so photorespiration doesn't really happen, but rather Calvin cycle will happen here because of the high concentration of carbon dioxide. And that is the way how C4 plants would adapt to high temperature. It concentrates the carbon dioxide in the bundle sheath, and it does so by converting the carbon dioxide into a 4 carbon molecule. So that's what's very interesting about the C4 plant. So you can see here, this is the mesophyll plant. the carbon dioxide enters here, and then the 3 carbon now becomes a 4 carbon. It now transfers the 4 carbon to the bundle sheath, where the carbon dioxide is released into the Calvin cycle. So carbon dioxide is indeed concentrated in the bundle sheath, preventing photorespiration to happen. Alright. Now, when we go to camp plants, another strategy to prevent photorespiration to be adapted in high temperature, camp plants, first of all, it's the meaning of CAM is crassulacean acid metabolism, and the reason for that is because this process was first discovered in cacti or cactus, which would be under the group of crassulacean plants. So what happens with camp plants? It also fix the carbon dioxide into a 4 carbon, but it does so very differently. First of all, the camp plants open their stomata during nighttime, and because it's nighttime, the temperature is very low, so this means water loss is lessened. So this is an adaptation by the camp plants to lessen water loss and at the same time be able to absorb carbon dioxide. So what happens here? After opening their stomata at nighttime and absorbing as much carbon dioxide as they can, during daytime, the stomata will then close to prevent more water loss, and then the carbon dioxide inside the carbon 4, the 4 carbon molecule, will then be released to the Calvin cycle. And this is what it looks like. So again, during nighttime, the carbon dioxide will now be fixed, producing a 4 carbon molecule, which is then stored in the vacuole. So at nighttime, it continues on storing and storing and storing a lot of the 4 carbon until the daytime comes. Once the daytime comes, the stored 4 carbon molecule, which contains the carbon dioxide, will now release the carbon dioxide into the Calvin cycle. So even if the stomata is closed during daytime, they were able, or the camp plants were able to store a lot of the 4 carbon. So there is no problem if the stomata is closed, because once the 4 carbon are released and the carbon dioxide would be released, it goes straight into the Calvin cycle, even in the absence of additional carbon dioxide, even if the stomata is closed. So that's what's interesting. Okay, again, just to summarize the comparison, they have the same process. The carbon dioxide is fixed into a 4 carbon molecule, both for the C4 and also for the CAM, and then afterwards the 4 carbon molecule will release the carbon dioxide into the Calvin cycles for both of them. But the main difference is that for the C4, the carbon fixation into the 4 carbon molecule happens in the mesophyll cell, and then carbon dioxide release happens in the bundle sheath. So it will be a separation of space, different cells. But for the camp plants, it will happen during the nighttime, and then the carbon dioxide will be released during the daytime. So it's a separation of time. And that's the end of this discussion.